So in this video we're going to talk about lipids, which is sometimes we call as fat. So what is lipid? Lipids are a family of biomolecules that have the common property of being soluble in organic solvent. Means these lipids are soluble in organic solvent. Organic solvent means non-polar solvent, but not in water. Because water is polar solvent, right? So always remember that fat cannot be solubilized in water because the lipids are non-polar in nature, so that's why they can easily be dissolved in organic solvent, which is non-polar solvent, right? And lipid also contain some fatty acid or some steroid nucleus, which we're gonna see what are these fatty acid and steroid nucleuses. They are soluble in organic solvent but not in water, like I already told you. And this lipid comes from a Greek word lipos, which means fat. So that's why sometimes we use the term fat instead of lipid. And why lipid is also important? What's the function of lipid in our body, on our cell? Because it is important for cell membrane, some vitamins, which is fat soluble vitamins, and some hormones, the chemical messenger of the body, right? For example, steroid hormones. So in this chapter we're going to talk about what are lipids, what are the different class of lipid, and what are their function. Now let's talk about the types of lipid. So lipids are characterized by their structure. Means when the lipid molecule get hydrolyzed, which type of product or structure they normally give. So some lipid, after getting hydrolyzed, means breaking of the lipid with the help of water, they release fatty acid. So that means that lipid is made up of fatty acid. That's why they are giving fatty acid as a product. And some lipid cannot be hydrolyzed. Means they cannot be broken down with the help of water. So that kind of lipid we put it under steroid section. So if you can see the difference in steroid, this is a fused ring, 4 fused ring together, right? And then the fatty acid structure is a long chain hydrocarbon with carboxylic acid group at the end, right? So fatty acid are the long chain hydrocarbon with carboxylic acid group at one of the end, right? So let's see which type of lipid, what are the different kind of lipid that give you a fatty acid as one of the product after they get hydrolyzed. So example are waxes. This is one of the type of fat, the lipid, fat, oil, and phospholipid. So these are the example that can give you a fatty acid as a byproduct after it get hydrolyzed, because all these lipids are made up of fatty acid and some other molecule. Steroid, on the other hand, do not contain fatty acid. As you can see this structure, it doesn't have any long chain of hydrocarbon with carboxylic acid end, right? And it cannot be hydrolyzed. Like I already told you, it cannot be hydrolyzed. So they are characterized by the steroid nucleus with 4 fused carbon rings. So steroid is normally characterized by 4 fused carbon rings, right? So this is how you can classify lipid, whether it contain fatty acid or whether they are hydrolyzed, or if cannot be hydrolyzed, that means that are steroid. So this is just an overview that we talk about from the previous slide. So we have lipid, then lipid can contain fatty acid in their structure along with some other molecule, and other lipids that cannot be hydrolyzed is placed under steroid, which has a 4 fused ring structure together. So there are four example or four type of lipid that contain fatty acid in their structure, that is made by fatty acid. So what are those lipids? Are waxes, triacylglycerol, 2 glycerophospholipid, and sphingomyelin is for sphingolipid. So these four type of lipid, when they get hydrolyzed or when you break the bond of these lipid with the help of water, you always get a fatty acid, because if you can see in their structure, they always have the fatty acid in the structure, right? So once you break the bond, you're gonna get fatty acid as a product. So that's why all these four types of lipid are placed under a group called fatty acid, right? So let's talk about the structure of the waxes. So what is waxes? Waxes is is a long chain alcohol with attached with fatty acid, right? Triacylglycerol is the attachment of glycerol, one of the alcohol, with 3 molecules of fatty acid. So we're gonna see each one of this structure later on, but right now it's just an overview. Glycerophospholipid again also have a glycerol, and over here you have 2 molecule of fatty acid, but the term is glycerophospholipid. So what does it mean by phosphor? You have, instead of the third fatty acid, that third fatty acid is normally replaced by a phosphate group, if you can see the difference, and that phosphate is attached with one more amino alcohol. And the last one is sphingolipid. So sphingolipid is made up of an alcohol called sphingomycin or sphingosine, and then this is different than all other glycerophospholipid and triacylglycerol because they have glycerol, but in sphingolipid you have sphingosine, and you have only one fatty acid and then phosphate and amino alcohol. So in this case the top fatty acid is get replaced by this sphingosine. So this is just an overview of all the four type of lipid that we're gonna talk about and that contain fatty acid in their structure, or they give fatty acid after get hydrolyzed. So let's talk about one of the problem that about all the concept that we just learned. So lipids are characterized by the presence of fatty acid or a steroid nucleus, which type is described by the following. A. Contain a fused 4-member ring system. So this should be steroid nucleus, because steroid nucleus is a combination of 4 fused member ring system, right? And this cannot be hydrolyzed. Option B is contain long chain carbon chain. So long carbon chain is, and at the end you have carboxylic acid, so it has to be fatty acid. And the third one is include carbonyl groups. So what is carbonyl group? Carbonyl group is C double bond O. This is your carbonyl group, right? In which type of lipid you can get carbonyl group, fatty acid or steroid? In fatty acid you can say this is a carbonyl group, right? In carboxylic acid, so this is also fatty acid, right? So this is how you can answer all this type of question depending on the structure that particular lipid have, right? So we already talk about this answer, why the first is steroid and other two are fatty acid. Okay, so now let's talk one more question on the same concept. Which of the following is not a characteristic of lipid? So which is not a characteristic of lipid? First, may contain carboxylic acid group. Yes, in a fatty acid you have carboxylic acid, right? In fatty acid, so this is a characteristic of lipid. May contain 4-ring structure. This is also correct. In steroid, one of the lipid contain 4-ring structure, right? So this is also characteristic. C is soluble in water. So in the first slide we just learned about lipids are not soluble in water or not soluble in polar solvent. They are always soluble in non-polar or non-polar solvent. or you can say organic solvent, right? So this is not correct, right? Mostly non-polar. This is also correct. We already know about this thing. Lipids are non-polar mostly. So our correct answer will be option C. This is not true. Fat lipids are not soluble in water, but it says soluble. So option C is our correct answer. So answer is C. So now let's talk about the fatty acid. We have been learning about lipids can be classified as a fatty acid containing lipid or a steroid containing lipid. So in this particular slide, we're gonna talk about a fatty acid. What is fatty acid? And what are the different types of fatty acid you can have in lipid? So you can say fatty acid can be of two types. This is Fa, fatty acid. So I can write fatty acid can be of two types, saturated and unsaturated. And what is fatty acid? Fatty acid is a long unbranched carbon chain with a carboxylic acid at the end. That means long chain of carbon with carboxylic acid at one of the end, right? We call as fatty acid. So fatty acid, like I already told you, can be saturated and unsaturated. We know what is saturated means. When this long carbon chain doesn't contain any double bond, so all are single bond, all single bonds in long chain of carbon. Wherein unsaturated means you might have some double bond in this long chain carbon of fatty acid, right? And this unsaturated can be divided into two: monounsaturated or polyunsaturated. Right? What is mono? Mono means you only have one double bond in the structure of fatty acid. Poly means you can have multiple double bond. Multiple double bond. So you can have two double bond, three double bond anywhere in the long chain of carbon of fatty acid, anywhere, right? So let's talk about now. So make sure you know this classification of fatty acid. What are saturated fatty acid? What are unsaturated fatty acid? What is mono and polyunsaturated are? Right. So now let's go back again over here. Fatty acid has normally 12 to 18 carbon atom longs. So that means how many carbon you can have in any fatty acid? Basically the maximum of 12 to 18 carbon. They are insoluble in water because we know carbon chains are hydrocarbon chain, and they are insoluble in water because they are hydrocarbons and they are non-polar. We already talk about in organic chemistry. Then we already talk about what is saturated means, that do not contain any double bond. Unsaturated means they should contain at least one double bond, right? We already talk about the classification. Now let's try to draw up some fatty acid, right? So we already know what are fatty acid. Fatty acid are long carbon chain with carboxylic acid at the end, right? So we can write any fatty acid something like CH3, then you have 10 times CH2, right? And then you have carboxylic acid at the end. So that means this fatty acid contain how many carbon? 1 plus 10, 11, 12. So this is a 12 carbon fatty acid, Fa, right? And you can draw the same fatty acid something like this. And then this is the line angle formula for fatty acid. So we already talk about all this condensed structure, line angle formula, right, in the organic chemistry part. So now let's talk about the saturated fatty acid first. So we already know what are saturated fatty acid, that contain only carbon-carbon single bond, that doesn't have any double bond, right? So there are a couple of examples: lauric acid, muriatic acid, palmitic acid, or stearic acid. All these four fatty acids are saturated. If you can see from the structure, you won't find any double bond. They are all single. Right? And then how many carbon you have in here? So you will see 12:0. So the first number tells you number of carbon, and the second number tells you double bonds. So in lauric acid, 12:0 means you have this lauric acid is made up of 12 carbon total. So if you can see 10, 11, 12 carbon. And how many double bond you have in this lauric acid? 0, means no double bond. Similarly for muriatic acid, if I write 14:0, what does it mean? This muriatic acid contains 14 carbon and no double bond. Right. So make sure if you see this kind of numbering 12:0, 14:0, or some random number 16:0, so you should be able to understand what does this number indicate, the first number and second number. Right. Now let's talk about the monounsaturated fatty acid. Monounsaturated means you will always have only one double bond in the structure. Right. So there are two examples: palmitoleic acid and oleic acid. If you can see the numbering again, 16:1. So 16 means carbons. So there are 16 carbon, and how many double bond you have? One double bond. So if you can see from this structure, you will see a double bond here, right? And there is only one double bond, and we have total of 16 carbon in this fatty acid: 5, 6, 7, 8 and 7, 15, and then 16. Right. Similarly for oleic acid, this fatty acid is made up of 18 carbon and only one double bond present in this structure, which is this. Sometimes we also call this monounsaturated fatty acid as MUFA, MUFA, monounsaturated fatty acid. Then we have polyunsaturated fatty acid, and sometimes we call polyunsaturated fatty acid as PUFA. And then poly means many. You can have multiple double bond in the fatty acid molecule. For example, linoleic acid, the numbers are 18:2. That means this linoleic acid is made up of 18 carbon, and there are 2 double bond present in the fatty acid. So if you can see, this is altogether 18 carbon, right? And you have 2 double bond. One is over here, second is over here. Right? Let's say the third one, arachidonic acid. This fatty acid is made up of 20 carbon, and you have 4 double bond present in this fatty acid. So this is the structure of arachidonic acid, and then you have 4 double bond: 1, 2, 3, 4. Right, so that's why this is polyunsaturated fatty acids are. And you can find this arachidonic acid in milk, egg and fish. So now let's talk about the cis and trans unsaturated fatty acid. Because if we know if the fatty acids are unsaturated, that means it contain a double bond. And the time we know it contain a double bond, then we can have 2 type of structures, cis and trans. Because double bond is very difficult to rotate, right? So that's why you have this kind of isomerism, geometric isomerism, cis and trans isomerism. So unsaturated fatty acid can be drawn as cis and trans, right? Because of the double bond present. So for example, this is a fatty acid, this is entire fatty acid, long chain carbon with carboxylic acid at the end, and then in any one place you will have a double bond, right? And because of this double bond, this structure can form cis and trans. So the same group is present on the same side, so this is cis. If the same structure is drawn something like that, where you have the same group on the opposite side, we call this as trans. So because of the presence of double bond, unsaturated at fatty acid can form cis and trans isomerism. And mostly all naturally occurring unsaturated fatty acids in the living organism have one or more cis double bond. So cis is and double bond are present. All naturally occurring fatty acid. Okay. So now let's talk about the essential fatty acid. What are essential fatty acids are? As the term is already saying you essential, that is important for our body. So humans are capable of synthesizing some fatty acid from carbohydrate and other fatty acid, but there are couple of other fatty acid that body cannot synthesize through any means. So that means body cannot synthesize sufficient amount of some polyunsaturated fatty acid. And what are those fatty acid that body cannot synthesize? Those are linoleic acid, linolenic acid, and arachidonic acid. So these three fatty acid you have to take from outside as in a diet, because body cannot synthesize these three polyunsaturated fatty acid within the body. So you have to take these three things in a diet, right? You have to provide all these three fatty acid to the body from outside. And because these polyunsaturated fatty acids must be obtained from the diet, they are known as essential fatty acid. Right? So you have to make sure you remember all these three fatty acid, polyunsaturated fatty acid, right? And you have to know what are essential fatty acids are. We're going to talk about some physical property of the fatty acid. So first we're going to talk about the saturated fatty acid. What are the physical properties are? As we know, saturated fatty acid contain only carbon-carbon single bond, right? And the saturated fatty acid fit close together in a regular pattern. If you can see, you have three fatty acid, 1, 2, 3, and if you can see, they can easily be fit perfectly right together in a regular pattern. So you can have easily fitted within other fatty acid, right? Which allows many dispersion forces between the carbon chain. So this is one carbon chain, this is second carbon chain. So if they come close together, that means you will have many dispersion forces. So dispersion forces is present between non-polar carbons, carbon and carbon, right? They are dispersion forces. So these dispersion forces are normally weak weak force of attraction. But if we have many carbon atom coming close together or many carbon chain coming close together, these weak force of attraction become important, right? As a result, the significant amount of energy and higher temperature are required to separate the fatty acid. Means if you have high or strong dispersion forces, means they are attracted strongly, and to remove or to break this strong dispersion forces, you need to have a higher temperature, right? Or some significant amount of energy. So that means saturated fatty acid always have a higher melting point and are usually solid at room temperature, because room temperature doesn't provide the required amount of energy that might break the strong dispersion forces, right? So that's why all saturated fatty acid are solid at room temperature, right? Compare the structure of the saturated fatty acid with unsaturated fatty acid. This is unsaturated fatty acid, right? So in unsaturated fatty acid, because of the presence of cis double bond, because of the presence of this cis double bond, the carbon chain is going to bend or kink like this. It's gonna kink. It's not a straight line anymore, right? And then because of this kink, the shape of this unsaturated fatty acid are normally irregular. As a result, unsaturated fatty acid cannot stack as closely as saturated fatty acids were, because they are irregular shape. If we come closer to fatty acid, they're not going to perfectly stack upon each other, and because the carbon chains are not perfectly stacked together, there will be a less dispersion forces present in unsaturated fatty acid, and because of the less stronger unsat less you can say or less, or you can say weaker dispersion forces in unsaturated fatty acid. So because of the presence of weaker dispersion forces in unsaturated fatty acid, the melting point of unsaturated acid are lower compared to the saturated fatty acid, because you need less energy or less temperature to break that weaker dispersion force within the unsaturated fatty acid compared to the saturated fatty acid, because the saturated fatty acid can be stacked perfectly. There is no kink or band structure, right? Where you can see in unsaturated fatty acid, they are not perfectly aligned because you have irregular shape. So that's why unsaturated fatty acids are liquid at room temperature. You can differentiate whether the given compound or given fatty acid are saturated or unsaturated, or whether they are solid at room temperature or liquid at room temperature. Let's try to solve this problem depending or based on the concept that we just learned. Consider the condensed structure formula of oleic acid. This is one of the fatty acid, right? And the structure is given. So the first question is asking why this molecule in acid? Because presence of carboxylic acid at the end, right? This is a carboxylic acid group, and then because of the presence of carboxylic acid, this molecule is an acid. B is how many total carbon atom is present in this oleic acid fatty acid. You have 1, 7, 8, 9, 10, 10 and 7, 17, and 1, 18. So you have total of 18 carbon in this molecule. Is this a saturated, monounsaturated, or polyunsaturated fatty acid? So how we gonna see? If we can see over here, all are single bond except this one. You have just one single double bond in there, and after that you have only single bond. So that means this is monounsaturated fatty acid. So this is the right answer for this problem. Is it likely to be solid or liquid at room temperature? Because this is monounsaturated, it is likely to be a liquid. Why? Because of the presence of double bond, there will be a weaker dispersion forces within this molecule, and that means you need less energy. So that means most probably it will be a liquid at room temperature. Would it be soluble in water? No, because this is a fatty acid and then it has a long carbon chain, and because of the presence of long carbon chain, this is non-polar structure, and that's why it cannot be dissolved in water because of the non-polar carbon chain, right? So the answer is already provided to you on this slide, but I already explained to you all the answer, but you can have a look over here too. This is liquid, they're insoluble in water. So I already explained about this problem on the previous slides. Now let's talk about the types of lipid that contain fatty acid in their structure. So we already know we have waxes and triacylglycerol. Out of the 4, these are the 2 that we're going to talk about right now. Right. Let's start with the waxes. What is waxes? Waxes are ester of saturated fatty acid and long chain alcohol. So and each containing carbon from 14 to 30, right? So it says this is an ester. So why this is an ester? Because this is fatty acid. What is fatty acid? Fatty acid is a long carbon chain of carboxylic acid, and it also says is a long chain alcohol. If you have a long chain alcohol, this is alcohol OH, and then you have long chain alcohol, right? Doesn't matter how many carbon you have in here. So what's gonna happen? Water is going to remove from fatty acid and hydrogen from the alcohol. Then what's gonna happen? You're gonna make an ester. So this is an ester, right? Over here you have fatty acid, other side you have alcohol. So this is a wax. So if you can see this structure, you have fatty acid, the same thing that I drew over here. Then you have an ester bond, right, or ester group, and this is the bond, ester linkage, and you have long chain alcohol. Right, so this is the structure, common structure for waxes, and where you can find this structure or this kind of lipid. Waxes are normally found in many plants and animals, and you can see or you can find natural wax on the surface of fruit, right, and on the leaves of the plant. You can find on the leaves of the plant, surface of the fruit, right? Why these waxes are present on these structure, fruit and leaves? Because they help to prevent the loss of water and damage from the pest, right? So this is like a covering on the top surface of the fruit and leaves, right? So this is the importance of waxes. Now let's talk about triacylglycerol, which is a second type of lipid that contain fatty acid in their structure. In the body, fatty acid are normally stored at triacylglycerol, right? So whenever you need energy, so the fat are normally stored in the form of triacylglycerol in human body, and sometimes we call triacylglycerol as triglyceride, right? So what's the structure of triacylglycerol? So the structure we're gonna see over here. Let's go to the next slide and then talk about the structure for any triacylglycerol. So in triacylglycerol, as the term is saying glycerol, so you have an alcohol which is glycerol, and glycerol is CH2OH, CHOH, and CH2OH. This is your glycerol structure, right? So you have 3 hydroxyl group attached with 3 carbon. So this is your glycerol. If you can see this from this structure, CH2OH, CHOH, and CH2OH. And then if you put 3 fatty acid side by side with the glycerol, what's gonna happen? So this is first fatty acid, this is second fatty acid, this is third fatty acid. So these fatty acid can be of same type, like in this example, each and every fatty acid has exactly the same number of carbon, right? And all are saturated fatty acid. So basically all 3 fatty acid that we are using to interact with the glycerol are same in this particular case, but this can be different, right? So out of the 3, 2 may be same, the third might be different. So make sure it always not the case that they will always have the same type of fatty acid. Okay, so when you have a glycerol and you have 3 fatty acid comes close by side, the carboxylic acid, the OH from the carboxylic acid of one fatty acid react with the H hydrogen from the alcohol from the glycerol is going to attach to remove as water, right, from second fatty acid, because fatty acid are long chain of carboxylic acid. So this is dehydration reaction, you can say, because you are removing water. So hydrogen come from the glycerol, OH come from the carboxylic acid part of the fatty acid, and this is also water. So what's gonna happen? You're gonna remove 3 water molecule over here, right? And what else left is going to connect together. So this oxygen is now going to connect with this carbon because other atoms are removed as water. So now you're going to see a new bond being formed. This is a new bond, and we call this is an ester bond because this is ester group. This is an ester group. Right. So in fat, we normally find an ester group when a glycerol and fatty acid combine together. They form an ester group, and the bond we call as ester bond. Right. So this is how this is triacyl glycerol. So this is how you normally make any triacyl glycerol. Like I already told you about, there may be a different fatty acid that can be used to make a triacyl glycerol. So if you are using different fatty acid with the glycerol, you're going to make mixed triacyl glycerol. So in this particular case, the first fatty acid that combined with the glycerol molecule to make an ester compound or group, that fatty acid is stearic acid. The second fatty acid that we used in this particular example is oleic acid, and the third fatty acid is palmitic acid. So in this particular triacyl glycerol, you have three different fatty acid that is attached to the glycerol molecule, right? So that's why we can say this triacyl glycerol is a mixed triacyl glycerol, right? If it was the same fatty acid, all three were the same fatty acid, that means this is a simple triacyl glycerol, not a mixed one, but this is a mixed triacyl glycerol. So triacyl glycerol can be used or is a major source of energy storage for animal, right? And basically most of the animal that goes into hibernate or that normally do hibernation normally store food or energy source in the form of triacyl glycerol. For example, polar bear. Right. Let's go. For example, polar bear, when he goes for the hibernation, it stores its energy in the form of triacyl glycerol. So whenever it needs food, it can just break the lipid or triacyl glycerol and get the energy that needed to sustain the life during hibernation. So let's try to solve this problem. Draw the condensed structure formula for a triacyl glycerol containing 3 molecules of mystric acid, which is 14:0. So that means you have to make one triacyl glycerol molecule that contains 3 fatty acid, and all 3 are same, which are mystric acid. So let's try this one. You have CS2OH. This is your glycerol. Right, and if you have 3 fatty acid, that means what is this 14:0 means? So you have 14 carbon and 0 double bond, right? So that means this fatty acid is a saturated fatty acid. So we can have 14. So this is OH, right? And then you can have 14. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12, 13, 14. Right? Again you can have OH. Then you can have one more fatty acid. So you have this fatty acid with 14 carbon with no double bond, and then at the end you have the carboxylic acid. Now what's going to happen if they react together? So you're going to remove water, and once you get to remove water, you're gonna make triacylglycerol. So this is your triacylglycerol. And this is the bond, ester bond, ester group, and this is your ester linkage, right, or an ester bond. So this is the structure that you need to draw when the question asks you about something similar type of problem to make a triacylglycerol. And the solution is already provided to you, but I already explained in the previous slide how you can make a triacylglycerol with a given number of fatty acid, right? So in the last slide I used line angle formula, but you can also use a condensed formula. You have one carbon, then you have 12 times CS2, right? So they just condense it into one parenthesis, and then we have CS3. So total of 14 carbon again. So this is also the right structure. So now let's talk about the chemical properties of the triacylglycerol. Which type of chemical reaction this triacylglycerol can show. The first chemical reaction that we're going to talk about the triacylglycerol is hydrogenation reaction. And we already know what is hydrogenation reaction: addition of hydrogen, right? This is hydrogenation reaction. So what's going to happen if you have any double bond? This is a general reaction. If you have any double bond, carbon carbon double bond, and if you put hydrogen in the presence of a catalyst, let's say nickel, so what's going to happen? One of the hydrogen going to attach over here, second hydrogen is going to attach on the other carbon that contain a double bond, and then this bond, one of the pi bond, is going to break to make that carbon carbon single bond. So this is happen over here. Now you're going to have single bond, right? So that means whenever you do hydrogenation reaction, you are converting an alkene to alkane, because right now every carbon has single bond, right? So let's apply the same triacylglycerol. So this is your one of the triacylglycerol, right? And then you have a fatty acid. You have first fatty acid, second fatty acid, third. So if you can see in this fatty acid, these fatty acids that is joined with the glycerol are monounsaturated fatty acid. All three are. Why? Because in all three of them you have one double bond, right? So how many hydrogen you need to convert all these three double bond into a single bond? So for each double bond you need 2 hydrogen, because each one of them need one hydrogen to remove one double bond, right? So if you have 3 of them, you need 6 hydrogen altogether. So that means 3 molecule or 3 moles of H2 molecule, right? So you should have H2, H2 and H2. So each H2 will go to each double bond and convert a double bond into a single bond. So if you can see now, you have all single bond present in this fatty acid. So that means you already did the hydrogenation reaction, convert a double bond fatty acid into a saturated fatty acid, right? Because we don't have any double bond in here anymore, right? All are single bonds. So this is how such hydrogenation work. Second reaction that we're going to talk about is saponification reaction. So what is saponification reaction? Is that reaction of fat when fat react with a strong base, and in this case we're going to use sodium hydroxide in the presence of an acid. So if fat react with a strong base, you form soap, and this process we call as saponification reaction. Right. So let's say how this saponification reaction work. So let's take this example. You have triacyl glycerol, right? And then you add NaOH, a strong base, with this triacyl glycerol. What's going to happen? So this is an ester bond. This ester linkage is going to break, right? And this hydrogen from the sodium hydroxide is going to bind with the oxygens. So you have 3 oxygen, that means you need 3 times of sodium hydroxide, right? Then you're gonna make what? CS2OH, CS2O, CHOH, CS2OH. So you're gonna make glycerol, right? And then the rest of the molecule, which is NaO, is gonna bind with carbonyl group. So you're gonna get O and NaO, NaO, and NaO, right? So this is a soap, a salt of carboxylic acid, right? This is a salt of fatty acid, and we already talk about the salt, how fatty acid normally make a salt, right? So this is sodium fatty acid, and the fatty acid is palmitic acid. So this is a soap. So the formation of soap we call as saponification. When you add triacyl glycerol in the presence of strong base, you always make a soap, which is a salt of fatty acid. Now we're going to talk about the type of phospholipid, which is phospholipid. So what are phospholipid? Phospholipid are a family of lipids that are similar in structure to the triacyl glycerol. And there are 2 types of phospholipid. One is glycerophospholipid and second one is sphingomyelin. So there are 2 types of phospholipid present, right? But let's talk about the structure of phospholipid first. So in phospholipid, you will have a glycerol. So basically we're going to talk about the glycerophospholipid, right, in this case. So in glycerophospholipid, you have exactly the same shape as triacyl glycerol. If you remember, triacyl glycerol has glycerol, and you have 3 fatty acids attached to it, right? This is our triacyl glycerol. But in phospholipid, especially in glycerophospholipid, you will see glycerol, 2 fatty acids attached to the 2 OH group of the glycerol, but the third hydroxyl group that's come from the glycerol doesn't have the third fatty acid, but it's replaced by a phosphate group, and that phosphate group is attached with an amino alcohol. So this is the only difference that you can find between triacyl glycerol and glycerophospholipid, that the third OH group from the glycerol is going to attach with the phosphate, not with the fatty acid, right? In triacyl glycerol you can have 3 fatty acid. In glycerophospholipid you will have 2 fatty acid, and the third fatty acid get replaced by phosphate and amino alcohol. So now let's talk about the glycerophospholipid first, and we already talked about the structure of the glycerophospholipid in the last slide, right? How they are different from triacyl glycerol. So you can have a read, but I already explained you how the structure is different. So from the previous slide, when we saw about the structure of glycerophospholipid, we found it out the third OH group from the glycerol is bound to the phosphate group. This is O fatty acid, CHO fatty acid, and the third OH group from the fatty acid is bound to the phosphate group, and this phosphate group is bound with alcohol, amine alcohol, right? So we're going to talk about this amine alcohol. What are the different types of amine alcohol that you can find it out in glycerophospholipid? So there are 3 amine alcohol that you can find that join with the phosphate group. So there are three of them. So one of them is choline, serine, and the third one is ethanolamine, right? So this amino alcohol can be one of these in glycerophospholipid, right? Because you have an alcohol group in here. So now let's talk about the types of glycerophospholipid. How many different types of glycerophospholipid we can find in the body? We have two different types. One is lecithin, second is cephalin. So if I say this is the type of glycerophospholipid, means they should have the same structure: glycerol, fatty acid, then you have fatty acid, then you have phosphate and amine alcohol, right? So the only difference between lecithin and cephalin, which is a type of glycerophospholipid, is in the structure of amine alcohol. If you can see in lecithin you have choline as an amine alcohol, where in cephalin you have ethanolamine alcohol, right? This is different. Except this, every other structure is same, because this is the same structure of glycerophospholipid. So depending on which alcohol is present as an amine alcohol in glycerophospholipid, you can have different types of glycerophospholipid. So this lecithin and cephalin are normally found abundant in brain and nervous tissue, also found in egg yolk, wheat germ, and yeast, right? So basically brain and nervous tissues are important because that are important for our body to function properly. So now let's talk about the structure of glycerophospholipid and about the polarity of this molecule, whether this molecule is polar or not, right? So now let's go to the actual structure over here. Then you can read about the glycerophospholipid, what is polar head, what is hydrocarbon tail, but I'm gonna talk about this when I'm going to talk about the structure. So now let's talk about the structure. This is glycerophospholipid because this is a glycerol molecule. So out of the 3 carbon of the glycerol, the first 2 OH group, as we know, is attached to the fatty acid, right? But the third OH group from the glycerol is going to attach with the phosphate group, and this phosphate group is going to attach with amine alcohol, right? This is your glycerophospholipid. So if you see it more carefully over here, so this structure is a non-polar structure of glycerophospholipid, right? Why? Because this part of the glycerophospholipid only contain fatty acid, long carbon chain, right? And long carbon chains are non-polar in nature, and then you have just carbon and hydrogen. But on the third carbon of the glycerol, if you can see, you have phosphate group and alcoholic group, amine alcohol. So this is a polar area. So within the glycerophospholipid molecule, you have 2 area. One is polar and second one is non-polar within the same molecule, right? So this polar area we can usually draw like this, like a head, we can usually say, and this 2 fatty acid chain we can draw something like this, like a tail. So this is the structure of glycerophospholipid. So if you see something drawn something like this, so that means is a polar head made up of phosphate and amino alcohol, and this 2 tail is form of fatty acid, right? So this is polar, this is non-polar, and this glycerophospholipid is the important component of cell membrane. We're gonna see or we're gonna talk about this glycerophospholipid mode when we're going to talk about the cell membrane, how the cell membrane is made up of, right? What kind of different component this cell membrane is made up of, right? But make sure you keep this in mind, the structure of glycerophospholipid. So that means if this is the structure, this polar head can bind with the polar molecule or can interact with the polar molecule like water or any polar solvent, whereas the tail molecule, because this is non-polar, it can only react with a non-polar solvent or non-polar molecule, right? So depending on where the glycerophospholipid is present, different part of the glycerophospholipid interact with different component of the solution, whether it's polar or non-polar. About the four type of lipid that contain fatty acid in its structure, which is sphingomyelin. So sphingomyelin is made up of sphingosine alcohol. What is sphingosine? It is a long chain amino alcohol, right? And you have, if this is an alcohol, the second, so you will have one fatty acid attached to this amino alcohol, which is sphingosine, and the third group is attached phosphate and amino alcohol. So how this is different than glycerophospholipid? How they are different than glycerophospholipid? Because in glycerophospholipid you have an alcohol called glycerol. This is alcohol that attach with two fatty acid, and the third group is attached as phosphate. Whereas in sphingomyelin you have the fatty acid, only one fatty acid, right? And the third one is still attached with phosphate and amino alcohol. So if you do hydrolysis of this sphingomyelin, how many fatty acid you can get? Only one. Whereas in glycerophospholipid you can get two fatty acid after breaking the bond, after doing the hydrolysis reaction, right? Okay, so where you can find a sphingomyelin, this lipid sphingomyelin are abundant in white matter of the myelin sheath. What is myelin sheath? Is a coating surrounding the nerve cell. So this sphingomyelin normally cover the nerve cell, and this sphingomyelin also increases the speed of the nerve impulse and insulate and protect the nerve cell, right? So this sphingomyelin lipid is very important to the transfer of or the speed of the nerve impulse that carry out all the information from the brain cell. So this is the structure of sphingomyelin sheath, right? So you have this fatty acid, this is your phosphate, and this is your amino alcohol. So now we're going to talk about another class of lipid that doesn't contain any fatty acid, which are steroids. So steroid that we already talk about doesn't contain any fatty acid or doesn't give you any fatty acid after hydrolysis reaction, right? And then they cannot be hydrolyzed, and there are two types of steroid that we're going to talk about. One is cholesterol, second is bile salt, right? So now let's talk about first about the steroid, right? What is steroid? Steroid is a fused ring of four carbon rings, right? So that's your steroid nucleus, and this steroid nucleus cannot be hydrolyzed, right? Right? And then you can number all these four rings as A, B, C, and D. So the most important steroid that we're going to talk about is cholesterol. Cholesterol is the most abundant and important steroid in our body. Right? Why? Because cholesterol can be found as a component in cellular membrane. You can find this cholesterol in cell membrane. You can also find in brain and nerve tissue. So this is why cholesterol is very important for the body, because that this cholesterol normally function in different part of the body, right, as a component. So where you can find where the body normally form cholesterol, in which part? So liver is the main area within the body that body used to synthesize cholesterol. So liver is the area where body synthesize cholesterol from with the help of fat. By using fat, you can say carbohydrates and sometimes proteins. So body use these three biomolecules to convert these biomolecule into a cholesterol. Right? So now we're going to talk about the bile salt. What is bile salt and what's the importance of bile salt in our body? Bile salt are synthesized in the liver from cholesterol, and it's normally stored in the gallbladder. And what's the function of bile salt in our body? Bile salt helps in absorption of cholesterol. If you can see over here, how bile salt is normally helps in the absorption of cholesterol. When you eat any lipid containing diet or cholesterol containing diet, that big particle of the cholesterol is very hard or difficult to absorb by the body, right? Because they are big in size. So bile salt act on this particle, and then they just break down this particle into a smaller particles, this cholesterol or lipid containing particle. And now you have the smaller particle that body can easily manage, and that's how this bile salt helps to digest or helps in the absorption of cholesterol. So that means bile salts helps in emulsification of cholesterol, means to convert a large globular particle of cholesterol into a multiple smaller particle that body can easily can absorb or work on it, right? So this is how bile salt helps in the absorption of cholesterol, by doing emulsification. So now we're going to talk about a special protein called lipoprotein, and we know this is a type of conjugated protein that we learn in the protein chapter. Conjugated proteins means this is a protein bind with lipid, and we're going to talk about what is this lipoprotein, what's the purpose of this lipoprotein in our body. So this lipoprotein helps in the transport of lipid. As we know, lipid is what? Lipid is non-polar, right? And it cannot be dissolved in polar solvent, like water, right? So we only learn about lipid cannot be dissolved in a polar solvent. They can only dissolve in non-polar solvent, means organic solvent, right? But if the body needs to transfer a lipid, for example cholesterol, if body needs to transfer cholesterol from one area of the body to another area, how they're going to transport? The only mean of transport is blood in our body, right? So the cholesterol has to go through the blood and move through the blood and go to the another part of the body, right? Another tissue, right? But the problem is blood is polar solvent, and cholesterol is a lipid which is non-polar. So it's very difficult for body to just move this cholesterol from one part to another part through blood, because non-polar cannot dissolve in polar solvent. So now body have a new technique or new ways called lipoprotein. They use lipoprotein to transfer a lipid, a non-soluble lipid, especially cholesterol, from one body part to another body part. So we're gonna see what a lipoprotein is. So lipoprotein is a lipid containing protein, means this is a globular protein, is like a circular in shape, made up of proteins and phospholipid. So they normally make an surrounding across a non-polar cholesterol molecule. This is your cholesterol molecule, right? And this cholesterol molecule is surrounded by proteins and phospholipid. Why? Proteins and phospholipids have polar head, right? Polar molecule, and they can dissolve in, or they can be transferred through blood, because blood is also polar. So this is how when the lipid molecule or cholesterol molecule gets surrounded by proteins or lipid, we call that surrounding area as lipoprotein. And when this lipoprotein containing cholesterol molecule goes into the blood think this is a polar molecule, and then that blood can easily transfer this lipoprotein containing cholesterol molecule from one area to another area, right? So this is how body normally transfer or move the cholesterol molecule from one part to another part with the help of lipoprotein. It's like a covering of protein and phospholipid, right? And this lipoprotein are soluble in water because the surface contain polar lipid and protein, right? And that can easily be dissolved in blood, like I already told you. So there are two classes of lipoprotein. There are two types of lipoprotein that body use to transfer the cholesterol molecule from one part to another part. One is low density lipoprotein, and the second one is high density lipoprotein. And we're gonna see what are their function. What is the use of low density lipoprotein and high density lipoprotein. Talk about what are the function of low density lipoprotein and high density lipoprotein. So low density lipoprotein carries cholesterol to the tissue where it can be used for synthesis of cell membrane, right? Or making some other thing from cholesterol. So LDL normally take the cholesterol to the tissue where it being utilized as a source of energy or the synthesis of some other molecule, right? Whereas HDL, high density lipoprotein, pick up the cholesterol, you can say pick up the cholesterol from the tissue to the liver, right? Where it can be converted into salt, and that can be eliminated from the body. So basically HDL normally carries the cholesterol to the tissue where the tissue need the cholesterol to make some other part, some other component of the cell, like synthesis of cell membrane. Whereas HDL, high density lipoprotein, carries out the extra cholesterol that the tissue has. So tissue tells, okay, I don't want this extra cholesterol. Then the high density lipoprotein goes over there, pick up the extra cholesterol, and then transfer or move that cholesterol to the liver, where that can easily be eliminated from the body. So that's why we call as HDL as a good cholesterol. Why? Because this cholesterol is getting out from the body, right? Whereas the cholesterol that's present in the LDL, low density lipoprotein. So if this low density lipoprotein carries high amount of cholesterol and it goes to the tissue where it need it, sometimes the tissue doesn't want that high number of cholesterol molecule. So if the tissue doesn't take all of the cholesterol that LDL molecule has, then the LDL molecule just deposited extra cholesterol that is not used by the tissue. The extra cholesterol will be deposited in the artery because tissue doesn't want this extra cholesterol molecule. So LDL, instead of going back, they just deposited extra cholesterol in the artery, right, where the blood normally flows, right, artery and veins. So if you have extra cholesterol keep on adding into artery, you're gonna get some plaque, right, or the arteries get saturated with the cholesterol. And then if you don't have enough space for the blood to move, then you will get some kind of disease, heart disease, because the blood is not moving smoothly, right, through these arteries, and sometimes you will get, or the people get, heart attacks because lot of cholesterol is accumulated within the artery that carries the blood to the earth or away from the heart, right? So that's why we call this LDL as a bad cholesterol, because it's not getting rid from the body, it just putting the cholesterol into the artery, and that helps to develop some kind of disease, right? So HDL is a good cholesterol, LDL is a bad cholesterol, right? Now we're going to talk about the cell membrane. What is cell membrane? What is the function of this cell membrane, right? Why the cell has this kind of membrane covering its surface? So cell membrane separate. What's the function of cell membrane? Cell membrane separate the content of the cell. This is your cell. This is your cell membrane, right? So this and this is outside of the cell membrane. This is outside of the cell. This is inside of the cell. So what's the function of cell membrane? The membrane of the cell separate the content of the cell from the outside environment or from the external fluid, right? And this cell membrane we can say is also semi-permeable. What is meant by semi-permeable? Means that the nutrient can enter the cell from outside. If you have any nutrient that the cells need, they can enter the cell, and as well as the waste product that the cell is making, they can go outside, right? So they can so that the nutrient and the waste product can go inside and outside, right? So that's why it's semi-permeable. And the main component of the cell membrane is made up of phospholipid, and especially glycerophospholipid. 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 And if you remember the structure of glycerophospholipid, you will see the head, and this is tail. This is non-polar fatty acid. Fatty acid tails, and this is polar head that contain phosphate and amine alcohol, if you remember from the glycerophospholipid, right? So we'll talk about the cell membrane. So cell membrane, as I already told you, separate the cellular content from the external environment, right? And it's the lipid bilayer. What is meant by lipid bilayer? It means you have two layers of lipid. This is the first layer of lipid, this is second layer of lipid, so that is bilayer, right? And then if we assume this is the inside of the cell and this is outside of the cell, so if you can see, this lipid bilayer has a special characteristic where the head, which is polar, is pointing outside, as well as the head for the lowest layer is pointing inside of the cell, right? And where you have these two fatty acid chain embedded within the cell membrane. So that means the center of the cell membrane is non-polar. Why? Because in the center of the cell membrane you always have just fatty acid chain. So that makes the center or middle of the cell membrane as non-polar, whereas the surface of the cell membrane is polar, right? So because it has a polar head which is made of phosphate, right? So why the lipid bilayer is formed this way? Because the outside environment of the cell, we have some blood and water, so that blood and water are polar molecule, so the head of the glycerophospholipid has to be on the outside because polar interact with polar. Similarly, the inside of the cell also contains some kind of water and polar liquid, so that the lowest layer of the phospholipid also has the head facing the inner side of the cell, right? Because polar interact with polar, and there is no polar liquid or molecule within the surface of the lipid bilayer. That's why all the fatty acid chain is present within the cell membrane, right? So this is how lipid bilayer work, right? So now I already told you about the cell membrane is a lipid bilayer where the outer layer and inner layer of the bilipid layer has a polar region, and then you have a non-polar fatty acid within the cell membrane. And why they have the polar head on the outer surface or the inner surface of the cell membrane? Because the outer layer of the phospholipid is in contact with the external fluid which may be polar, and the inner layer is in contact with the inner content, means inner content of the cell, right, which may be polar too, like water and some kind of polar liquid, right? And most of the phospholipid in this lipid bilayer contain unsaturated fatty acid. So the time I say this cell membrane is made up of glycerophospholipid, so that means it made up of fatty acid, if you remember the structure. So this fatty acid in glycerophospholipid that made up the cell membrane is basically containing more unsaturated fatty acid, right? They are not saturated, and because of this presence of this unsaturated acid, you will have a kink in the carbon chain and to make this double bond, right? And then because of making of this cis double bond in the fatty acid, the phospholipid molecule cannot fit close together, and if they cannot fit close together, lipid bilayer is not rigid but it's flexible, right? And because of this flexible nature of the cell membrane, there is a model we call as fluid mosaic model. Fluid means it's flexible, right? And we already talk about why the cell membrane is fluid, because of the presence of unsaturated fatty acid, right? Okay. This cell membrane also have carbohydrate present. They also have some proteins embedded into the phospholipid bilayer, right? And they have some proteins like I told you, right? So the lipid bilayer is made up of many different components. The main component is phospholipid, but you have carbohydrate as well as protein. Right? So the lipid bilayer, like I already told you, is contain protein, carbohydrate, and cholesterol, and because of the unsaturated acid that make the cell membrane fluid-like rather than rich. Right? We already talk about this thing. And what's the purpose of protein and carbohydrate on the cell membrane? Because it can interact with the hormones and neurotransmitter that have all the information that cells want from the outer environment. Okay, so now we're going to talk about the transport through the cell membrane. Like I already told you, cell membrane is semi-permeable. So there are three different ways cell can transport substance across the cell membrane. So the first way of transport is diffusion, second one is facilitated transport, third one is active transport. So what is diffusion way of transport, or we also call as passive transport? Passive transport means moves the particle or substance from higher to lower concentration. So this is just a diffusion. For example, if you have a high concentration of any molecule on the top, and if you have less concentration of the same molecule on the bottom, so by diffusion the molecule goes from high concentration to low concentration, right? This is just simple diffusion. Second one is facilitated transport, which use protein channel to increase the rate of diffusion. So if you see some molecule need, so let's say this is your lipid bilayer, right? And if you have any particle that want to enter the cell through the cell membrane, this is polar in nature. Is it possible for this particle to move through a normal cell membrane or in a normal way? No, because the inside of the cell membranes is non-polar. So when it crosses the head, there will be a non-polar, so it cannot cross the non-polar part of the cell membrane. So that's why cell membrane has a special type of channels called protein channels. So if it is protein, this area is polar, so the molecule can easily pass through these channels and goes inside the cell rather than going through the non-polar part of the cell membrane. So that's why this is called facilitated transport. Means you are facilitating the transport of one particular molecule across the cell membrane to increase the rate of diffusion, right? The third one is active transport. Active means where you need energy to transport the molecule. That means which moves ion against a concentration gradient. What is meant by the against? Let's assume the same example that I told you over here. If I want to move the molecule from bottom to up, is it possible to go? No, because the right way of movement is from top to bottom, like I already told you, from high concentration to low concentration. But if I have to go in opposite way, we have to provide some energy. We have to give a push so that the molecule can go from lower concentration to higher concentration, which is against the law, right? So we have to use energy to do this active transport, right? Let's try to do this problem with the concept that we just discussed. The transport of particle across a cell membrane from high concentration to a low concentration. So this means the particle is going from high concentration to a lower concentration, right? So this is simple diffusion, because this is the way normally particle moves from higher concentration to a lower concentration, right? The answer is diffusion.